So we're going to move up the evolutionary ladder away from cetaceans. Down the IQ scale. So we're going to start with this seal, sea lions and fur seals and otters, or others, doesn't the same thing. And we're going to start with a student here at UBC, Mariana Diaz Gomez, who's going to be talking about some of her work with fur seals.
Thank you, Dave. Hello, everyone, and hello, everybody at the overflow room. So my name is Mariana. I am a master's student at the Marine Mammal Research Unit, and today I'll be talking to you about part of my thesis work here in the Marine Mammal Research Unit. And I decided to label my title The Middle Child of Nutrition because micronutrients sometimes are not always well locked. So what does it mean to be the middle child? You know, sometimes they do their own thing. You know, they get overlooked. Sometimes they get pushed to the side. They get ignored. And so this is what I feel that has happened to micronutrient digestibility in seals. And so I think that the middle child is important. Why is this? Well, animals, all animals, have specific requirements for lipid and proteins. So this is what when I say micronutrients, I specifically mean lipids and proteins. So let's say a female that is lactating, she wants to make fatty milk for her pup to grow strong and healthy, and so she needs that high amount of lipid. Let's say a young pup that wants to put down growth tissues to grow bigger, then he needs high protein in their diet. And so this is why these micronutrients are important for animals. And also, the micronutrient digestibility is directly linked to the amount of energy that the animals obtain, because lipids and proteins both provide energy. Energy that can then be used for swimming, for thermoregulation, and so on. And so for my research, I wanted to answer 3 important questions. The first one is, how efficient are fur seals at digesting both of these micronutrients? And also, what are the dietary elements that may affect the digestibility efficiencies? And I wanted to test the theory that a mixed diet will provide a greater micronutrient return than single species diets. To answer these questions, I had the joy to work with 6 northern fur seals at the Vancouver Aquarium, as well as all of the marvelous staff there. These animals were subject to 8 different diets. To create these diets, I used 4 prey species that I either combined to create mixed species diets or fed them in single species. I also collected scats, because who doesn't love doing that? And so with this, I was able to extract the lipid and the protein of both, what went in and what went out. And by a differential, I was able to calculate the digestive efficiency of these macronutrients. So some of my results really quick. It shows that both lipid and protein digestibility are very high. So then you could probably think of northern fur seals as your dream juicer. Like, it gets the most out of what they're eating. However, dietary protein significantly affected the digestibility of both of these, but in different ways. Lipid digestibility decreased with increasing protein content, whereas protein digestibility increased with increasing protein in the diet. And I showed that the mixed diets do not provide a better macronutrient return than single species diets would. And so why should we care about the middle child, right? Well, the first seals at the Pribilof Islands are currently suffering a population decline, as well as some other very important pinnipeds in the area. And it has been hypothesized that they are potentially nutritionally stressed. So what is nutritional stress? It means when there's a mismatch between what you consume and what you actually require to burn as fuel throughout the day. And so it's possible that the first seals that have been constantly nutritionally stressed, they may be absorbing nutrients in a different way and potentially not as efficient as the first seals that have been very well kept, healthy, and with lots of vet care at the aquarium. And so they could potentially be compromised in their nutritional budgets. And so if they're not obtaining as much as they need and they're not eating high quality food, then this could be detrimental for the whole population and not just the individual level. And with that, I would like to thank everybody that has helped me throughout my entire research, and I'll be happy to take any questions. I'm clapping for myself.
Do you have a question for Mariana? Uh-oh.
Hi. Yeah, I don't know who said that. Oh, I might have said that wrong.
I might have heard it wrong too.
Oh, so I found that high protein content in the diet is impeding lipid digestibility. So it's mainly because fish have this inverse relationship, so that fish that are high in protein are usually low in lipid. And so the higher the protein in the diet, then the lower the lipid digestibility. So it might be an effect of the relationship, just as because there's lower lipid to absorb, then it may be lower. But we found that it was statistically significant. So that's what I'm saying, that sometimes prey quality is more important, because prey quality, we think about it as in terms of fatty fish. And so fattier fish, you get the most out of it. There was a question higher up. No?
Nope.
Oh, there you go. Hi. Yes, I knew it. Right, the question was if there was an efficiency difference between invertebrates and vertebrate fish. Well, I guess, yeah. No, I did not find a difference. However, the squid we used was quite fatty, and we were only able to use it as in combination with herring. I was never able to use it just 100% herring diet. Squid, sorry. But we did not find that it caused a significant difference compared to the other diets. Yeah, thank you.
Thank you. Katrina Nikolich is now going to tell us something about harbor seal vocalizations.
Thanks. All right, hi, my name is Kat. Excuse my voice. I promise I am in fact a female human. Just experiencing a cold right now. So what I want to talk to you about is the real speed dating version of some of the work I've been doing from my master's thesis down in Western Washington University. That's in Bellingham, Washington, but all of this research was done actually in Canada. So harbor seals, as you're probably aware, are one of the most common species of marine mammals in this area. The most common you'll find in the Strait of Georgia. I'm looking at their breeding behavior in particular, so the patterns of their breeding behavior. They breed during the late summer and early fall in this area. During that time, the males will produce underwater vocalizations. They will establish territories in the water, usually around where the females are hauled out. And then they'll produce these underwater vocalizations both to deter other males as well as potentially to attract females. So this harbor seal, the breeding call is called a roar. It was first described in Monterey Bay, California about 20 years ago. So I don't know how many of you actually knew that harbor seals make underwater sounds before I started talking about this, but it is still a fairly new area of research. This breeding call has been heard in a number of populations throughout the harbor seal's global range. We believe this is a call that's used to actually sort of show physical and sexual fitness to other individuals. This is what it sounds like. Okay, maybe the sound system has been turned down. I'm sorry, guys. That's okay. If you can imagine somebody breathing into a microphone, sort of like, I'm not going to do that and get the microphone wet, but that's basically what it sounds like. So it's a very diminutive call. It's a little bit hard to pick out. It's actually easier to see on a spectrogram than it is to hear in real life. Now, of course, harbor seals can't be calling all the time. Breeding calls have to be a part of their energy budget, which means that they have to be feeding, they have to be resting and doing other things. So understanding the timing of when they make these breeding calls on a daily and yearly basis can give us an idea of the environmental and biological constraints that are acting on these animals while they're calling. So we know that in harbor seals, call timing can be influenced by tide, and that has to do with hollow-out patterns, which makes sense because as the tide rises, you have more seals in the water that are potentially calling to one another. Time of day has been shown to have a difference, as well as the time of year, because they do have a discrete breeding season. They don't tend to vocalize outside of that season. So this has been shown in other populations. However, the breeding behavior and the vocal breeding behavior of harbor seals has never been studied in the Pacific Northwest, despite numerous harbor seals in this area. Something else that I think is really interesting to look at in terms of these environmental factors is anthropogenic noise. As we know, and as we've seen earlier today, anthropogenic noise does have some behavioral effect on other marine mammals, especially with regard to their vocalizations. This has not been looked at in terms of harbor seal vocalizations. So what I wanted to look at is what environmental factors, including ambient noise, affect the vocal behavior of animals in the Strait of Georgia, which is an incredibly noisy region, as you saw earlier, really heavily trafficked. Is it the time of day or the tide that I wanted to look at, as has been shown in other areas? I also wanted to look at the ambient noise level. So this is a very open-ended question. These are three things that I wanted to look at in particular. My study site was Hornby Island, chosen because it just has a really high density of harbor seals that are accessible visually from shore. So if I zoom in here, this is southern Hornby Island at a place called Heron Rocks. This is where we did our research. So we had the hydrophone positioned in the middle of the rocks that was recording continuously from June through September 2014. And then we took visual observations from a nearby site on shore. Once I had all this data collected, I started to go through, and a real brief version of my analysis, I'll show you a picture of it later, is that I went through and took 20 random days from the breeding season, and I counted the number of vocalizations that I heard in each hour of those 20 days. This is the most striking trend that I found. So this is the average number of calls per hour over time of day. The shaded areas are when it was dark. So right away you can see that these harbor seals preferred to vocalize almost exclusively at night. So of course the next question is, why would this be? So we can probably all agree that it's energetically favorable for males to call at times when they're most likely to be heard. You're not going to bother calling if nobody's around to hear you. So the first thing I wanted to look at is, does this have to do with tide? Do those 20 days that I picked just happen to be when the tide was highest and most seals are in the water at night? I had a look at our visual observations from during the day, and it turns out that we saw exactly what you would expect. So there was more seals hauled out and less seals in the water around low tide, which is indicated sort of in the middle there, and then the opposite happening at high tide. So this is what you'd expect to see. However, from our statistics, the number of calls per hour was not related to tidal height at all. So if that tidal haul-out pattern persisted overnight, we can also infer that the harbor seals were not calling more when there were more seals in the water. But of course, there's other things at play here. So another thing I wanted to look at, which unfortunately there isn't a lot of information about, is foraging patterns. So were the seals just foraging at night and therefore more active and vocalizing at night? There's not a lot of information in this area about when these animals are are foraging. We do know that harbor seals are adapted for nocturnal and diurnal foraging, and that they're opportunistic, especially in this area. Harbor seals will eat what they can, when they can, as much as they can. So it's likely that the seals are foraging both during the day and at night. I can't confirm that, and that's something that I'd really like to look at a little bit further with this. But of course, being heard doesn't just mean the number of animals that are around you. You can be in a crowded bar, and there can be as many people as there are in this room, but if the music is blaring really loud, they're still not going to hear you. So one other thing that I wanted to look at was ambient noise level. We know that the seals preferred to call at night, where it was significantly statistically lower noise than there was during the day. More interestingly, the number of calls per hour, regardless of the time of day, increased as the noise level decreased. Now, of course, time of day and ambient noise level were really highly correlated, so I couldn't speak to each one of these individually, but there's some suggestion that these seals are calling when there's less noise. We also know that the kind of noise that we are looking at, mostly anthropogenic vessel noise from outboard motor vessels, overlaps with the harbor seal calls over about 1 kilohertz, so the higher component of these calls, the higher frequency component of those calls has been shown to be really important in actually conveying the dominance of a particular animal. So this important information could be masked by the boat noise. So it's possible that the males in this area are calling at night to actually avoid having the information in these calls masked by vessel noise. This is by no means conclusive evidence, but I think it's a really interesting thing to look at, considering that harbor seals in this area are doing just fine. They're obviously having no trouble reproducing, but there are areas in the world where you don't have this diel pattern of boat traffic, where you actually have equal boat traffic during the day and the night. I'd like to look at these areas as well as areas where harbor seals are in a population decline to see if this could actually present some sort of conservation concern. So I'll leave you with that for now. I'm happy to talk to you about this later. I'm also going to be, shameless plug, I'm taking a poster to the Marine Mammal Conference in San Francisco this December, so I may see some of you there as well. So thanks very much. I'd like to acknowledge a number of the people that made this possible, especially my collaborators at JASCO Applied Sciences, which provided all of the equipment as well as the expertise for this study. And this is a picture of me during my analysis. I'll take any questions.
Any questions for Kat?
Hi.
Front.
Yes. Yeah, so what was asked is there is a visual display on the surface, so sort of flipper slapping that's sometimes associated with these calls. And he's wondering if I had seen or if I had heard this underwater in association with the calls. The surprising answer is no. Not any more than I heard it associated with other kinds of behavior. We heard a lot of flipper slapping during the day, but it wasn't associated with those calls, which is different from what's been seen in other areas. It's usually been really highly associated. And similarly, when I was hearing a lot of the roars underwater, it was maybe 50-50 that there would be flipper slaps associated with it. There was really no correlation between them, which I found to be interesting. Yeah.
Much, Kat. Okay. Our next speaker is Aaron Purdy, who's going to be talking about some diving physiology.
Hi there, everyone. Yes. So I am Aaron Purdy. I am the new master's student here at with the Marine Mammal Research Unit, the aforementioned newbie in this title slide. So starting off with the newbie, he's that guy over there. So I'm from Alberta. I've had very relatively little experience with ocean environments. So I started out, I just wanted to come up with a question of what I wanted to do my research on. So I went through the literature, doing a lot of studying, and I finally came up with this. How do you science? No, obviously not. I'm looking at something a little bit more intellectual than that. Best part about this picture is that isn't even a textbook. That's a script from a musical. So, what I'm interested in is looking in the aerobic dive limit of sea lions and pinnipeds. So the aerobic dive limit is how long an animal is capable of staying underwater using only their oxygen. This is really important to help determine how long they're actually capable of foraging. So because of that, this doesn't have just connections to physiology, but also to their ecology and conservation as well. I specifically am interested in looking at what happens when pinnipeds exceed this aerobic dive limit. So they are capable of diving past this point where they are no longer using oxygen, but instead they're going to start building up lactate in their body. And this is associated with a lot of different costs. First cost, obviously, is going to be recovery time. They're going to have to stay at the surface to process that lactate in their body before they're able to dive again. If they're going to be staying at the surface for a long period of time, they're obviously going to be decreasing the amount of time they have to forage. Other costs that I'm interested in looking at are the respiratory muscle and lung function. It's possible that with an increase in lactate in the body, there's going to be some cost to the respiratory muscles. Maybe they are no longer able to work as efficiently, and therefore they're not going to be able to have as quick of a recovery. So once again, affecting that recovery time and affecting their foraging. I'm also interested in looking at some other physiological changes, particularly in the blood, possibly looking at things related to oxidative stress. So where I will be working, I'll be working out at the Open Water Laboratory in Port Moody with 4 trained Steller sea lions out there. I've just got the opportunity to go out there a little bit recently, and it is fantastic. I'll just show you kind of what the setup they have out there is. These sea lions are expertly well trained, and we are capable of manipulating their voluntary dives by providing fish at depth down to their relevant foraging depths. And then when we want them to come back up, we stop feeding them, and they will come back up into the surface, and they'll surface in this respirometry dome, where we'll be able to measure their metabolism. So my study plan, what I'm hoping on doing is 2 main things. First one is defining the aerobic dive limit. The aerobic dive limit actually hasn't been specifically measured in many species. It's only been measured in 5 marine mammals in total, only 2 pinnipeds, I believe. And so that's because it's very difficult to actually get these blood samples from these animals, and that's the only way of measuring this aerobic dive limit. Luckily, with the trained sea lions that we have at open water, seeing that they're so expertly trained, I'm going to be able to get some voluntary blood samples and finally define what the aerobic dive limit is. There have been many different calculations and estimates of what this dive limit might be, and this will be able to help kind of finalize as to what it actually is. How we'll be doing that, manipulating the dive duration with food provided at depth, and then using a combination of respirometry and voluntary blood samples. The other thing that I'm interested in looking at, as I said before, is the cost of exceeding this aerobic dive limit, specifically in these three different things: the recovery time, the respiratory muscle function, and their blood physiology. I haven't been able to get out there too much, but I'm hoping that I will get a lot of time to spend with these sea lions. I have lots of people to thank and lots of people I will be thanking in the future. And if anyone has any questions, feel free to come ask. Yep. Yeah, so question of what are some of the potential things I'd be looking at in the blood physiology. So there are other metabolites that might be building up in the body through catabolism and things like that, and also possibly looking into antioxidants as well that shows that sign of that oxidative stress. Any other questions? Awesome, thanks so much.
Our next speaker, who you will quickly detect is not from around these parts, is going to be speaking about some research that's happening in the other part of the world.
Thanks, Dave. Hi, I'm Monique. I'm a visiting researcher from Sydney, Australia, and I'm working with the Marine Mammal Research Unit for a couple of weeks here. But I thought I'd switch it up a little and not talk about a BC marine mammal and instead give you guys some insights into some of the physiology from the Australian guys that I've been working with. So just a little bit of background. Oops, I've gone too far already. So I'm working with 3 species of otariid, or otariid, as you say in North America, I believe. The Australian fur seal, the New Zealand fur seal, and the Australian sea lion. All of these guys were hunted to near extinction in the late 1800s and have since experienced different rates of recovery. The Australian fur seal has generally recovered. The New Zealand fur seal is expanding its population and is starting to move into territories that were previously held by the Australian fur seal and the Australian sea lion. But our poor old Australian sea lion over there is endangered and is in decline at the moment. And these different rates of recovery are really interesting for management purposes, and we're wondering if physiology can maybe explain some of these things. So fur seals and sea lions have to travel really long distances and dive to great depths in order to obtain their food. And these have large energetic costs. But these costs differ across taxa and within species, particularly across the different life stages. So the aim of our project was to identify how the energy requirements might differ between and within the Australian otarid species. So how do we go about doing this? Aaron actually just gave a pretty good rundown of it, but I thought I'd show it to you instead. So this is Ronnie. He's a New Zealand fur seal housed at Taronga Zoo in Sydney, Australia. And at the moment, we're taking a measure of his standard metabolic rate, which is where he sits in the respirometer until we get a baseline measure. After that, we ask him to swim laps of the pool to measure his active metabolic rate, at the end which he'll return to the dome, and we can get an estimate of active metabolic rate. And as we know how long they swam for and the size of the pool, we can estimate speed, which we use for our cost of transport measurements. And cost of transport we're using in this case because we're really interested in how much energy they have to expend to get to their foraging grounds. We conducted 318 trials with the 3 species that I already mentioned, and in my study sample I had 11 seals. So some results. We found that there is indeed a decline of metabolism with age. I was lucky enough to have 5 female Australian sea lions in my study. There is actually 2 here, even though you can't really see it. But the great thing about this was they were they're the same age, around about the same weight, and they had the same metabolism. So I was very excited because it validated my results. Then we wanted to have a look at how the metabolism might differ across the different cohorts that I had. So I had some quite unique cohorts, including doing some of the very limited amount of physiology on males, which hasn't really been done before. But as you can see here is looking at the adults, we get roughly about the same metabolism across those 3 adult species. But when we add in the subadults, you can see that they're consistently higher than the adults within each of those cohorts. And then if we add in the active metabolic rate, this effect is exacerbated. Then we wanted to look at cost of transport. So the open squares in each of these graphs are the subadults of the species, and then the closed squares are the adults. And the main things to take away from this are that, again, we can see how much higher the cost of transport is for subadults as opposed to the adults. But also that when we look at the adults, we can see that the optimal swimming speed, and speed is along the x-axis here, is between about 1.5 and 2 meters a second. And this was also good for me because it validates some of Dave's research, or my research was validated because it's around about the same time as, around about the same speed as Steller sea lions, sorry. So what did we find? So we found that age matters. So as seals get older, their metabolism is declining, which can have really important management implications, as this may be a vulnerable group that we may need to start looking at. And fur seals and sea lions reach physiological maturity really late. In the fosid seals, they actually get weaned a lot earlier than otariids do. And this can have this can have implications for their management as well, because if they're maturing really late, it means that they've got higher energetic costs for longer. So species did not really make a difference, and it seems as though metabolism remained constant across the species. So what does this actually mean? I've got to do some more research. So I'm currently looking into how season affects the animals, also looking at how gender affects metabolism as well. And hopefully I'll see you guys soon to tell you some more about it. Thank you.
For Monique. Okay. I'm just making sure somebody's in the audience and hasn't snuck off. Aaron, are you here? Okay, I'll make your way. So I'm giving the next talk, but as you'll see, no, get down here. So I decided. So I run. Sorry, my name is Dave Rosen. When I'm not doing this, whatever this is. Most of my time is running the captive Pinniped research group that UBC runs in collaboration with the Vancouver Aquarium. And I've been doing it now for, this month marks 20 years. So yes, I started when I was 7. And so, no, you don't need the paper. Calm down. Aaron's not sure why he's up here yet. So the aim of our Captive Pinniped Research Group is it's one of the tools that we use at the Marine Mammal Research Group to use science to inform management and conservation policy. So there are certain things that we can do in a laboratory setting with marine mammals that we cannot do with animals in the wild. So we can answer certain specific questions that managers want answered. So understanding things like physiology, testing technology, things like that. The program was established by Andrew Trites in 1993. Currently, we have 4 female Steller sea lions and 5 northern fur seals at the Vancouver Aquarium, and we have another 4 at our open water research station that Aaron's going to be working with. A lot of our research has been examining something called the nutritional stress hypothesis. There are a number of species, both marine mammals and seabirds, that are undergoing dramatic declines in the North Pacific and Bering Sea, and we're trying to investigate whether these population declines are related to changes in the availability or the type of prey that they are feeding on. And so a lot of our effort in the last 20 years has been trying to address the possibility from a physiological basis of whether the nutritional stress hypothesis has a basis. As I said, I've been doing this for 20 years. Look at that handsome guy, but I'm the one on the right. More hair, less bags under the eyes, and my knees work. And so Andrew said, well, why don't you do a summary of 20 years of research in 5 minutes? And I said, nope, that's easy, too easy. 20 years of research in 5 minutes. So I'm actually going to do it in the form of a series of haikus. Now, because my haikus sound a little bit like really bad fortune cookies, Aaron is going to provide the translation. Now, I want to emphasize, Aaron up till now has no clue what is going on. So we're going to start. Not all fish equally good. Contrasts more extreme after passing through seal's guts.
Different prey are different in nutritional quality. Low quality prey is even less valuable once it has been digested.
Eat more fish when it's required, but sometimes your needs may exceed your digestion.
While individuals can increase their food intake to compensate for differences in prey quality, the amount of low quality prey required may be more than they can physically process.
Sea lions need food to thrive. The seasons differ. Shortfalls worse at certain times.
The food requirements of pinnipeds vary dramatically during the year, leading to seasons where they are more susceptible to food shortages.
Poor diet effects are worse in certain seasons for survival and healing.
The physiological consequences of episodes of nutritional stress and the ability to recover are both highly seasonally dependent.
If energy needs are met, quality is moot. If not, prey fat matters more.
If sufficient energy is obtained from prey, prey quality has no direct impact on health. However, if insufficient energy is obtained from prey due to decreased availability or quality or increased requirements, low prey quality has additional negative physiological effects.
Less fish to catch in the sea changes dive patterns. Costs go up, intake goes down.
Decreases in prey density principally alter dive behavior, increasing energetic costs and reducing overall foraging efficiency.
20 years of research shows nutritional stress could certainly cause declines. Thank you, Aaron, for your assistance. Oh, you got one more.
Okay. There is a reasonable physiological basis to support the contention that population declines in marine mammals in the North Pacific and Bering Sea are related to changes in type or abundance of prey species.
Thank you very much. I'd like to thank Aaron nobody should legally be allowed to put their new master's student through something like that. I'd also like to thank particularly the Vancouver Aquarium, who has been our partner for 22 years in this one-year project. And of course, all of this research was only accomplished by a vast team of trainers, veterinary staff, research assistants, students, graduate and undergraduate students, and of course, money from our bedrock funding agencies, both in the US and Canada. I'd like to thank them all for their continued support.
Thanks, Dave.
No problem. Any questions? Any questions at all?
I have a question.
Oh.
Dave, did you forget that haikus are 575, not
No!
757?
No, it's 757. Oh. I have just invented a new haiku form. Oh. Okay, yeah. Yes, and if you can say something in 17 words, why do you need 3 pages? That'd be the take home message.
You wouldn't have been able to do what you did with
Oh, yeah, I could. No problem. Okay. Well, getting us back to reality. Oh, sorry. Yes, Steve. Well, yes, we have, but only on a gross level. So when we talk about changes in prey type, if they're losing mass, if they're not getting any energy to have sufficient mass when they're on a low lipid diet, as we are all told in the media, they're actually losing more lipid mass, proportionally more, which is good for us but bad for marine mammals. We've also had some studies that have actually looked a little bit at things like oxidative stress and telomere integrity, but they're not particularly my projects. But obviously we can't, and when you've got valuable research animals, there's only so far you can push as far as negative health effects when you've got a controlled population. So getting back to more serious science, or any science at all, is Ben Nelson, also from UBC, is going to be talking about potential interactions between harbor seals and salmon.
Thanks, Dave. My name is Ben Nelson. I'm a PhD student here at the University of British Columbia, and my PhD research involves probably the first rigorous quantitative assessment of the harbor seal impacts on Chinook and coho salmon here in the Strait of Georgia. And the impetus for this research is probably this really impressive change in abundance of harbor seals that's occurred in the Salish Sea since they were federally protected in the 1970s. So in 1970, there were probably fewer than 5,000 animals that called the Strait of Georgia home. And as recently as 2008, it's been estimated there are about 45,000 animals. And our neighbors to the south have seen similar increases in the Puget Sound. And coincident with these really impressive increases in abundance, two of the most important salmon species for British Columbia, Chinook and coho, have seen really marked declines in productivity and abundance. And these trends have led some scientists and a lot of fishermen to speculate that harbor seal predation might be impeding the recovery of these two really important salmon species. So to date, most of the research that's been done has occurred on interactions between harbor seals and adult salmon that are returning to their natal streams to spawn. And what we really sought out to establish was the impacts that harbor seals might be having on salmon at the juvenile stage. And the reason for that is a lot of the research has suggested that the primary limiting factor for salmon populations right now occurs somewhere in the first 3 or 4 months of the salmon's life at sea. So I won't bore you with the nuances of the modeling and the mathematics used in these analyses, so feel free to grill me during the break about the details or the assumptions that we used. And I'll jump right to the results that used some really interesting DNA-based diet studies that our colleagues at University of British Columbia just finished up. So for coho and Chinook salmon, we found that about 55% of natural mortality for coho can be attributed to harbor seal predation. And for Chinook, about 45% of natural mortality can be attributed to harbor seal predation. These peak mortality rates occur right when coho salmon hit the water, or the salt water, in April and May, and a little bit later for Chinook in July. And when you combine these mortality rates with the timing of predation, what we've estimated is that about 2/3 of coho salmon smolts are lost to harbor seals within the early marine stage, and over half of Chinook salmon smolts are lost to harbor seal predation in the first marine stage. So this line of evidence seems to suggest that harbor seals are indeed having pretty important impact on the productivity and marine survival of these two very important species of salmon. And what's interesting is that the peak mortality rates that we've estimated occur when these salmon smolts fall into a very narrow size range. So somewhere between 100 and 200 millimeters is when the seals seem to be targeting these smolts. And that might have implications for how hatcheries are managed, and you know, there's a lot of interesting questions that come out of that observation. So what we'd like to do is use these mortality estimates and consumption estimates. We'd like to use them to improve the assessment models for salmon. So a lot of the assessment models that are currently used to manage salmon populations, they make this kind of unrealistic assumption that mortality rates are stationary or don't change over time. So we'd like to incorporate what we found into these models to hopefully improve their predictive ability, as well as to evaluate different management scenarios like altering hatchery releases, altering different harvest strategies for coho and Chinook. And these models should also, hopefully in the future, should be used to be able to evaluate different ecological scenarios. So for instance, if there was an increase in transient killer whale predation on harbor seals, could that increase in mortality on harbor seals possibly facilitate recovery of Chinook and coho salmon in the future? And yeah, with that, I'll take some questions. Yes. Yes. Yeah. Sure. So the question was, you know, to deduce these mortality rates, you have to have some estimate of initial smolt abundance in the spring when all these fish are released. And so since a large contingent of these smolts are of hatchery origin, we have very good estimates of the number of hatchery fish that are released in a given year. And from abundance surveys being conducted by DFO, basically that give us a fraction at any given time of wild fish to hatchery fish throughout the summer, we can essentially scale that up using that proportion to estimate the number of wild fish that are out there at the same time. That's the assumption that we're making, is that there is basically that the catchability of wild fish and hatchery fish are the same, and that's definitely an assumption that would probably need more empirical studies to defend. Thanks.
Our next speaker, Malgosia, is going to be speaking about an event that happened in California that many of you may have heard about in the news.
Oh, I don't see anything. Hello, everybody. My name is Malgosia. I'm a marine mammal trainer at the Vancouver Aquarium, and I work with Steller sea lions, northern fur seals, and David. And today, I will be talking about my trip to California in March of 2015, where I was helping with rescues of the stranded California sea lions. So the problem, where? In California, like I said, mostly between the counties of Santa Barbara and San Diego, there were hundreds and hundreds of starving, dehydrated, malnourished, and sick sea lion pups washing ashore. There was such alarming number of them that NOAA declared it an unusual mortality event. Suspected reasons for the stranding was El Nino. You all are aware what El Nino is, so I'll say it in my understanding. So there's warm winds from the south that are not counterbalanced by cold winds from the north. Water temperature rose from between 0.5 to 2 degrees Celsius, and that has pushed good fish like anchovies further away from the areas where sea lions feed. So the mothers who gave birth to their pups, instead of going and foraging for 2-3 days, leaving that unattended pup on the island, had to go further and further away to find prey, and they would leave the pups for 8 to even 9 days unattended. And those little pups were starved, they were frustrated, they started going into the sea without any ability to either forage for themselves or actually even eat if they caught something. And they got weak, malnourished, and started washing ashore. That's the basic mechanic behind this. Numbers, in 2011, I'm quoting NOAA on this one, so pretty accurate numbers. In 2011, there were 82 sea lions pups stranded in California. 2013 was when NOAA announced the first unusual mortality event, over 1,200 animals. And now get ready, 2015, 3,340 animals stranded up till June 1st of 2015. You may wonder, will this hurt the California sea lion population? The scientists don't think so. The California sea lion population is estimated at 300,000 animals. So this is barely over 1% of the population. So even though it's so horrible, it will probably not affect the population of the wild animals. NOAA asked 3 rescue centers in California, in Sausalito, San Pedro, and Laguna Beach, to assist with the California sea lion strandings. And I got a chance to go to Laguna Beach location, and you can see it on the picture. The red barn is where the rooms for indoor sea lions were, basically the sea lions that were too weak to do anything else than lay down and not die. And the side pools were for the animals that were stronger enough to go in the water and could get out of the pool. The pools in front of the building are the pre-release pools where the sea lions were housed permanently before the release, and that's where they were competing for the food that was delivered into that pool. So that was the best pool, lots of noise. Rescue stages, intake. On intake, you go to the beach, you pick up the sea lion, bring it to the rescue center. Immediately, the vet team assesses the condition of the animal, the weight, the heart rate, and whether or not to euthanize it on the spot. Unfortunately, there was a cutoff weight at the bottom as well. If the animal was less than 17 pounds, it would be euthanized right away. Surely, because the number of sea lions coming into the place, that sea lion would take up a spot that you could rescue 3 other sea lions who were slightly higher at the start weight. And then if the sea lion was progressing to the next stage, it would be given sub-Q fluids with lots of dextrose in it, so basically sugar to boost their energy. And we started feeding them. First feeding is a tube feeding, which you need 2 people. One person is restraining a sea lion. The second person is leading the tube into its stomach and pumping a fish formula, which is made of herring, blended herring and Pedialyte. The next stages of feeding, as the animal grows and learns more how to eat, would be force feeding the actual fish. Then the animal, like this one on the picture here, would eat from the bowl. And that was a very exciting moment because this is when they start eating on their own. And the last stage of feeding would be feeder pools, where the animals were delivered few buckets of fish dumped in at the same time, and they had to compete with one another. Obviously, that would happen in a wild. There is a school of fish, and you have to compete with other sea lions to get the food in. Throughout the feeding stages, there's also health and development assessments. So every 3 days, every sea lion would be put on scale and weighted. So we had to know that they're growing at a healthy rate. And of course, you observe the body condition, the skin condition, the energy levels. The facility had 125 animal capacities. As you can imagine, there was a lot of animals to be observing every single day. And then the sweetest part of the rescue was the release. Bittersweet, because you kind of get attached to them, even though you shouldn't. They go back to the ocean. The release weights for 2015 were actually slightly lower than the regular season. So in a regular season, the sea lions would be released at 65 pounds, which is 25 kilos. And in 2015, because of the volume of animals, they wanted the animals to go faster. So they were releasing them at 22 kilograms. Trainers to the rescue. So I had this amazing opportunity from the Vancouver Aquarium that I got to go and help out with the rescue efforts 4 trainers and 6 vet techs from the Vancouver Aquarium headed down south, and we volunteered our times over there. There were trainers, and you may know this girl, that's Andrea. She's a pathologist from the East Coast. And a lot of scientists, trainers, vet techs headed down there, and we all put the boots on, rolled up the sleeves, and we all did the same. We helped rescue those pups. There was no task too small or too big for us, really. On location, daily duties. There was 3 things you did. You cleaned, and then you prepped food. And that was basically 12 hours every single day of this. But those were amazing learning opportunities, especially for me, I felt. I learned how to restrain a sea lion by myself using just a towel. I got to go to a rescue and pick up an animal on location. I assisted in tagging of an animal pre-release, and I also got to observe a necropsy, which for me was super educational. This is how you carry a restrained sea lion. So you wrap it nicely in a towel, and you carry it just like this. And how does this compare to being a trainer? It's totally opposite of what I do. So like I said, I should not have get attached to the animals because you don't want to build a relationship. You don't want the animals to get used to you. You want them to avoid humans because that's how they should act in the nature. They should not be coming up to us and expect any sort of interaction. The question is the crisis over? So the sea lions are not stranding anymore at this point. However, Guadalupe fur seals are. And northern fur seals, they have over 80 northern fur seals in the rescue centers right now, and they still keep washing ashore for suspected same reasons. And for more information, you can always visit marinemammalcenter.org, and they talk about how you can help with a crisis and how you can help to spread the word and help with the actual rescue. So thank you.
Any questions for Malgosia?
So the question is, how malnourished were the pups? A healthy sea lion pup from San Miguel Island at 3 months of age is about 17 kilos. The pups that were washing ashore they were washing ashore from January, but they were from the breeding season of the previous summer. So at 7 months old, they were between 8 to 12 kilograms. So they were below their birth weight. So super, super skinny. Yes? So the question is if there were other reasons for emanciation than just being stranded, right? Because from the necropsy, actually, that I got to observe, the veterinarian told us that some of the animals he observed had parasites that are found in the grass. So they were actually going to areas where they're not supposed to be going. So not sitting on a sandy beach, but going inland and getting parasites that were found in the grass. And they had it in their lungs, and that would kill them as well. Well, at that age, they shouldn't be inland that much. They should be still on that island where they were born, basically, because they're not traveling with mothers yet at that age. Yes. So the question is about the survivability. English is not my first language, of the animals. They assess it at 50% at this point. So the way you look at it, 3,000 stranded, 3,000 could have died, 50% of them got to live. Some of the animals were equipped with GPS tags, and they actually are being tracked and confirmed still alive. Yep. One more question? A question was if females were affected as much as males?
No, adult females.
Adult females. I was there for a week, and we had one intake of an adult animal that was about 2 years old and was malnourished. And other 124 that were at the facility were pups. Great, thank you.
Thank you. Our next speaker can barely speak, but he's going to tell us a mystery story.
well, I may be whispering my story. About a year and a half ago, I was approached by lawyers working for two tribes in the Olympic Peninsula, the Quileute and the Quinault. And they've been in court for the past 35 years trying to get back their treaty rights. When they signed the treaty in 1856, it ensured them the right of taking fish at all usual and accustomed grounds and stations. The trouble has been that their fishing rights were not being respected. So they were in court against the state of Washington and also the Makah tribe, their neighbors to the north. You can see on here the territories of the Quileute, the Hoh, the Queets, and the Quinault. And the Makah had come down from what is now known as British Columbia and had pushed the Quileute further south. And so there was essentially a battle to establish what was the normal and usual fishing locations of people at the time the treaties were signed. On this map here, you can see the bathymetry, and the locations on red are showing you the Indian midden sites. So there are a few European accounts that can be looked at to get some insights, but for the most part, the challenge is to try to interpret the middens, or they would just be in the garbage pits of where the remains of animals were put. So for us, we're asked to tell the court, so in the 1850s, what marine mammals were present. And of course, nobody was really collecting much data. There's a couple of naturalists did some wrote a few books, give us some insights, but they really want to know what was out in this 20 to 40 miles offshore. And so we can recognize that these are really highly productive areas. In fact, they're so productive, I think we can think of them as being the Serengeti of the ocean. There are herds of animals that are moving by, and I'll tell you a bit about that in a minute. We know there's 12 species of marine mammals that are occurring further than 20 miles offshore, and I think it's fair to assume that the behaviors of animals today is the same as animals as in the past. We know from the middens that all these species have got some remains found in them. But when you look at, okay, except the human diver. Yeah, I saw you there picking it out. But the remarkable thing is that, and there are first-hand accounts of fin whales being brought ashore that had been killed. But when you look at the bones that are in the middens, over 80% of them are from only one of these species. And it's from this one down here. They're from northern fur seals. And what's remarkable is that these northern fur seals, they don't come to shore. They only breed in 4 locations that we knew of in the 1850s. And to get them, they would have had to go offshore, 30 to 60 miles offshore to get them. So there I'm showing you where the 2 tribes that we were involved with were located. Here are the 4 breeding locations: the Pribilof Islands, Commander Islands, Robben Island, Krill Islands. The animals breed there, and we know the Pribilofs do migrate, and they migrate down off of California and go back up. So presumably the source of the bones was coming from the Pribilof Islands. We know a lot about the movements because after Alaska was purchased, a huge pelagic sealing industry was started, where boats were sailing out of Victoria in particular, taking native hunters with them to do the killing, to capture the seals and to get the furs. These 4 breeding sites, rookeries, well, not quite like this, but like this except for the catwalk, because you can't walk in among the animals. It's that dangerous, but the animals are very tolerant. You can be over top of them, but should you fall in, they will kill you. They're on really remote, foggy, wet, and dangerous spots. The dangerous part just being with them. The interesting is that none of these 4 sites ever had humans living on them. They are a pelagic species, and they never come ashore except in the summer to breed. The basic family structure, you have these bulls, which beef up from an average of 300 pounds to 450 in the breeding season. But when you compare to the females that only weigh a mere 80 pounds, pups at 11, that means the dad is about 6 times bigger than mom. And it's always amazing when you ask kids this, can you imagine if your dad was 6 times bigger than your mom? And they kind of go, wow. But what we know from the history of the sealing, the pelagic sealing, is that only females were being taken. Many of those females were pregnant, but there were no bulls in amongst them. The bulls stayed further north from the Pribilofs. But here's the big mystery. When you look in the middens, it's got the skeletal remains of unweaned pups, juveniles, and bulls. And there were some historical documents that led to a belief that these seals had come from somewhere else, and possibly a local rookery, except that there's no record of them ever having bred on land in Washington. So out of that came a paper published by a group out of University of Victoria. And you see the title of it here is The Cape Flattery Fur Seal, an Extinct Species of Calorhinus. Sorry, commercial break. And so they had concluded, sorry, they had concluded that this had to be an extinct species, but they also drew some other conclusions, and they said, and the reason it wasn't seen on land is because it bred on beds of kelp, which of course for a biologist is just not possible, that seals need to have some sort of solid substrate, whether it's ice or rock, but they have to be out of the water to give birth or the pups will die. So this is what the courts were faced with, and I spent 2 days on the stand with really grueling cross-examination to come to terms with the fact of what I'm calling here a fairy tale was not possible, but I had to come up with a better explanation. So first off, when you look at the historic bones, there's a paper published that looked at the DNA. It came long after Susan and Becky's paper. And what they found is that based on the genetics, there's only one species of fur seal. The ancient seals are carbon copies of the modern seals. So they weren't a different species. And they suggested the Bering Sea was this refugia that allowed them to migrate and therefore be captured. But another piece of data that was really hard to come to grips with, and that was stable isotopes. These are essentially chemicals that are laid down in the bones, and they tell us something about what the animals ate and therefore where they were. And so here I'm showing you for nitrogen, and you'll see the blue line down below shows for the Pribilof population, it starts high because they're pups, and drops as they move from essentially eating mum, which is by drinking the milk, to moving down to eating fish, and then we see an adult signature coming back up again. So if those ancient ones were all from the Pribilofs, they should look like the blue, but you see in red that they had a very different signature. So let's just catch up to my story. The red ones are up here, and this just doesn't fit with them having come from the Pribilof Islands. It just doesn't make sense. So it does make sense when you see that where they're congruent is the pup stage and also out there at the adult stage, and suggests that maybe these animals were from a different population that we forgot about or wasn't known in the 1850s. So here we've got the 4 sites in 1850, and we've had a few things happen since then. A new colony was started in 1968 in San Miguel, another one up here in Bogoslav in 1980, and now a new one at the Fairlands in 1996. So now instead of having only 4 places in the world, we now have 7. Genetically, they are panmictic. They're all carbon copies of each other, but they're really remote sites. So pay attention to the Fairlands, because what we didn't know at the time, but now know very well, is that there was a lot of sealing going on. Fur seals are being killed and traded in the late 1700s to 1800s. And the Russians also had a sealing station. So we did a lot of research looking at log books and historic records. And they did indeed have a sealing station. But by 1841, that population had been extirpated. There were no more fur seals there. bring in some more modern data. These are from weaned fur seal pups that have been satellite tracked. And showing here these three colors, red is St. George, yellow St. Paul, and green Boguslav. You can see that those animals are coming out of the Bering Sea, going into the North Pacific. And these are young ones and not coming close to shore. But look at this magenta color. Those are from San Miguel. And we have other data also for now adult females. And so we clearly have two distinct foraging ranges from two distinct populations, although there is wandering too, like that one in magenta that took off north, thinking it was Alaskan. So here we have our current systems. And the Alaska current system and the California current system. And here in BC, we're at this transition zone. We tend to think a lot about Alaska and being part of that, but really part of two different systems. And it appears then that the fur seals that are born in California are retained within this California current system. And that would apply also to the bulls. And so this is then the explanation for these unique signatures and why the entire family was found within these middens. They came from a different population with some adult females from Alaska being mixed in. And just very quickly here to wrap up, the only place to get these seals is offshore. And there are a few reports, first-hand accounts of having gone out. One person, his name is Morgan Roth, reported getting up early in the morning, sailing out because they had sails on their canoes, sail out and killing with spears fur seals out 30 miles offshore. I mean, it's remarkable. Because I know when I first started looking at this, I was quite skeptical about, I couldn't paddle out there. And then the more we read, the better we began to understand and appreciate the amazing navigation skills of the First Nations people. Just a few other quotes here. This is Hal George, born 1894, and he wrote that the seal hunters, or he was recorded saying seal hunters sometimes took off at 2 a.m. They wanted to get out to what was called Oxpasta, blue water, the place where the ocean really gets deep. Through this case, we got to work with linguistics experts, and there's an amazing number of words to describe the ocean and different parts of it, and color was one of the key ways to distinguish different places. Beatrice Black, born 1890, described, my brother used to go out as early as March, go out in the ocean, way out to get some seals. 2 or 3 months he'd go out. They risked their lives going way out, 40 miles out in the ocean in an open canoe, 3 men in a canoe. Sometimes they'd be loaded with about 5, 6, or 10 seals. And this is Chris Morganroth, who I referred to earlier. Seal hunting by coastal Indians takes place during these 2 months, April and May, in the 100-mile stretch of open ocean between the mouth of the Creeks River and Cape Flattery. Here, the seals approach nearest to land, the line of northerly migration being about 30 to 50 miles offshore. When out far in ocean, another quote this was from Arthur Howettle, when out far in ocean and unable to make shore, canoes were anchored thus. The sails were laid flat on the water, stones were tied to them on each end, and canoe made fast to the sails and stones by means of a rope. What fits so well with the biology also is, so we've got these sealing implements, and some of these are in museums, others just from drawings at the time, the spears that were used. A few photographs also from this time showing how it was done. A seal club, and of course they could spear them, and they were going out, so in the morning, the seals that had fed all night, because they had to wait for the fish to come up to the surface, would be sleeping, and then they could be speared and brought on and clubbed. And so, conclusions then, and this is some of the seals are brought back to shore, and they're all fur seals, you can see that really clearly. And so it appeared then these Farallon and Pribilof Islands were refugia for northern fur seals. Humans never found these islands, and they provided regular and dependable food and furs. And here's a bit of speculation at the end. I suspect that the fur seals, it may well have happened in British Columbia, where the continental shelf is really narrow and comes in. The people first encountered the fur seals. And this technology and knowledge, they were out there in the blue water, in that Serengeti, was passed down to tribes up and down the coast. And so I suspect the First Nations may well have been responsible for the development of these incredible seafaring abilities. Thank you very much. Boy, that was tough.
Any questions for Andrew? Okay.
Oh, and I should just add, and they won their case. And they couldn't believe it after 35 years fighting it that they won it. And a lot came down to the evidence about marine mammals.
Okay, our next speaker is Roy Mulder, who's going to be talking about trying to organize scientists, I think.
Yes.
Yes, good luck with that.
So I haven't got any pretty graphs or pictures or anything else. I'm here to talk about kind of a cat herding expedition. Essentially, we're facing the UN Convention for Biological Diversity coming up in 2010 or 20, and we're still talking about it, but we're barely talking about it. Now, I'd like to do a little show of hands. How many people in this room are directly working on any programming to create marine protected areas of any sort? How many people in this room? Okay, so maybe what, about 3% of this room? How many people in this room would like to see marine sanctuaries in Canada? Okay, so the vast majority of us would like to go there. Now, the challenge we have is Canada has probably one of the biggest coastlines of any country in the entire world, and we protect 1.3% of it fully. That's the number. A lot of people don't realize it. They hear a lot of terms that say marine sanctuary. You hear terms that say marine reserve. You can look at something like rockfish conservation areas. There's a lot of different terms and a lot of things that imply protection, but there's very few that actually are full protection. I mean, we can look at larger areas, we can look at smaller areas. I do a lot of, I'm not a scientist, but I go to a lot of the conferences and everything else, and the takeaway that I've taken from that is, if we're going to protect the ocean and we're going to create protected areas, we have to protect probably 20 to 30% of the area, and large areas and large swaths. This isn't happening. Canada has just gone through a very dark time in advocacy and creating protections. We've seen some things happen. I've been part of a group protecting sponges. You think it's hard to protect charismatic animals like whales and dolphins? Try and protect sponges. And this is the thing. And what happens is, you know, we talk a lot about protecting things, but we do it from a species point of view, and we see a lot of speciesism out there. There's a lot of conservation groups that are out there. They're the shark group, and they don't want shark finning, and they want to protect sharks. We have another group, they want to protect pinnipeds. We have another, well, mind you, I don't know if there's any groups protecting pinnipeds. Is there? Maybe, but nonetheless, what happens is people get very focused in one area. But if you take a look at any single species, and if anything else, the takeaway from this room today is the fact that you walk out of here and you know everything's connected to everything else. When you're protecting one species, you can't say, oh, I'm going to protect one species and not protect the habitat and the creatures that it survives off of. And so, you know, in a world today where all of a sudden krill has suddenly become a really neat health food. So this is now going to threaten whales. You know, you have to look at how it's all connected. So how are we going to connect all these organizations? So we got 3% in this room. These are the people I want to talk to, who by all means talk to me after this. How do we join us all together to work towards one common goal? And I think that that's how it has to be done. If we all appreciate that we're looking at all these various different facets of it, but for us to actually become inclusive and get that 10% of Canada, we have to be talking about that now. It's 2016 almost, and we're saying 2020 we're going to set this area of Canada aside, but I'm not seeing the dialogue happening yet. And so I'm here standing here today saying, we have to start creating this dialogue and saying, it's all well and good to spend your lifetime as a scientist studying these things, but I've got friends who are working on vaquitas right now, and they're sitting there and they're going to watch what they've been studying get studied until the last one in the species goes. And this is what we have to be concerned about. And if we're going to protect everything, we've got to start talking to each other, and we have to find a common, simple goal. And I think that that's where we fail. I think we get too much into the complexity of things. we also don't realize just how much is gone. 9/10 of the world's big fish are gone. That's a fairly common number that everybody's using. Recent studies have seen that, we've almost lost 50% of our fish stocks from 1970. I mean, those are very frightening statistics, and especially when the creatures you're studying are the very ones that are surviving off of these other creatures. So there's an incentive for all of us to join together and say, let's create this area. Where is it, and what does it look like? And how can we do it in an effective way so that we're protecting all the species rather than any individual one? And this is where it becomes very difficult because they all seem to hang out in different areas. I can only look at my own little backyard. I dive every weekend. I'm in House Sound almost every weekend scuba diving. I've been diving 42 years. I used to go out and I'd see a school of rockfish. I am hard-pressed to say that I can find a school of rockfish in House Sound anymore. I can tell you where there's one, but if I did, it'd go away. Then if we speak towards that aspect of it, rockfish conservation areas were invented so that we would protect rockfish, but they allow all sorts of other forms of predation. So if you're going to allow seining in a rockfish conservation area, you're going to get bycatch, and it's kind of defeating the purpose kind of thing. If you create these protected areas without enforcement, and that's the other thing, we can talk all we want about protection, but if we're not talking about the enforcement aspect of this, it's not going to work. I mean, the RCAs, when they created them, it was a great idea, but then next thing we discovered was they put them onto charts. And the next thing that happened after that was poachers got a hold of the charts and they became targeted. So all of a sudden we've created these protected areas, but and we've identified the areas, which is probably the biggest mistake we made, and then it became a poaching area because there's not enough enforcement. And I talked to the people, who work at DFO and they're working on it. I think we need to make sure that any plan that we have includes enforcement, but we need as many organizations to join together. So I would like to ask if anyone's interested in joining me and my organizations with this task. That's Marine Life Sanctuary Society, Canadian Marine Environment Protection Society, World Cetacean Alliance. I'm members of all three. By all means, look me up, find me, help us. We can use it. Any questions?
Are there any questions for Roy? He's got lots to talk about during the break. Okay, all right. Okay, so again, we got a double presentation on the next one. Whale Trail, BC, Bailey and Donna.
Hello, everyone. My name is Bailey, and I'm with the Marine Mammal Research Program at the Coastal Ocean Research Institute at the Vancouver Aquarium. Got to work on that. So I want to tell you a little bit today about how the BC Cetacean Sightings Network is working to increase land-based data collection by helping to expand the whale trail throughout BC. Now, some of you may be wondering what the whale trail is, and I'm actually here with Donna Sandstrom, the director and the founder of the whale trail. It's a land-based whale watching initiative, but I'm going to leave it to her to fill you in on that a little more in just a moment. So at the Sightings Network, we receive sightings from a great diversity of sighting platforms. But one thing that we noticed recently is that we were actually receiving a fair amount of sightings from land. So we saw in the last 5 years that we actually received 14% of our total sightings were from land, despite the fact that most of our outreach is targeted toward mariners. We also noticed, or we actually have 6% of our total database that is from land. So seeing this pattern, we reached out to the whale trail, and they were very keen to collaborate. So we created a steering committee, and a couple people who are actually here today helped us to create a list of sites where people were having beneficial land-based whale watching experiences. And also, we looked at Sightings Network data as well to see where people were already having these sightings from land. So once we had identified those locations, we moved towards creating panels like this. So this is one example of our panels. So you'll notice that it encourages people to report their sightings. It has response network information, and it also has species It shows what animals you're most likely to see in that area. So when people are looking at this panel, we hope that it will help them make a positive identification, which can help our data collection when they're reporting their sightings to increase their confidence. So thanks to Mountain Equipment Co-op, we have 5 panels that are going up starting next week. Thieves Bay on Pender Island. We also have the Wild Pacific Trail in Ucluelet, the Shaw Ocean Discovery Center in Sydney, Lighthouse Park in West Vancouver, and then in Telegraph Cove as well. And we also have 28 panels that are installed or being installed throughout the Southern Gulf Islands, and those ones are just a condensed version so people can take a look at that and report their sightings to us. So moving forward, the Sightings Network hopes to continue to help expand the whale trail throughout BC, but we also want to take a look at these areas where we've installed the panels and see if we're actually getting greater amounts of sightings reports here. And if so, we would like to use this as a measure of observer effort, perhaps, for some of our effort-corrected work. So that's a very quick summary of what we have going on in BC. I'm going to pass you off to Donna.
You realize you're getting 2 for 1, and I have to talk twice as fast. I have sheer terror of the chicken. Okay. So I'm going to use my time to tell you a little bit today about the prologue, the whale trail story. And the whale trail story actually begins in 2002 with a whale called Springer. How many of you have heard of Springer? Oh, I'm so happy. That doesn't happen everywhere. So Springer is an orphaned orca who showed up near Seattle, where I live, in 2002. And it was through her calls, it was determined that she was a member of the Northern Resident Community, the A4s. She was 300 miles away from home. There was virtually no way her family would find her or that she would find her way back to them. I'm going to tell a wonderful, wonderful story. I'm going to make it very short. We, the community, persuaded NOAA Fisheries that if she was healthy enough, she should have a chance to go home. And the scientific community agreed that she was a good candidate for reintroduction. Those 2 things combined to lead to the first ever successful orca reintroduction in history. Springer was transferred home on a donated catamaran to the north end of Vancouver Island, and her pod came to pick her up less than 24 hours after she was returned. It was a complete conservation success. This is Springer getting welcomed home, waiting for her family. And thank you, Graham, for the great photo. As I'm sure you're all aware, this is Springer. Springer had her first calf 2 years ago. The calf survived its first year, and its name is Spirit. And that's totally appropriate because it was the spirit of that project and the collaboration that drove it. We had to get Springer home, we had to work together, reach across every possible boundary, and put the whale's best interest first. And in that spirit, we founded the Whale Trail. In 2005, the southern resident killer whales were listed as endangered by NOAA Fisheries. The threats are all human-caused: lack of prey, toxin accumulations, and stress and noise from vessels. I was stunned when I read in a Fish and Wildlife report this population could go extinct in as few as 100 years. That is a minute in time, and we are living in that minute. So I believe it's up to us. The future of these whales who have been here for tens of thousands of years, they're in the palms of our hands. So we founded the Whale Trail with many of the same team members who had seen Springer safely home, including NOAA Fisheries, People for Puget Sound, the Whale Museum. We added the Department of Fish and Wildlife. Again, you'll see that it's a collaborative effort. We found a simple but powerful piece of common ground, which is we wanted to promote shore-based whale watching. We wanted to build awareness of the southern resident orcas throughout their range. And we wanted to encourage stewardship. All conservation begins with awareness. We wanted to let people know who these whales were, where they lived, that they were endangered, and that we all had a piece in their recovery. We started with 16 inaugural sites in Washington State. To be on the Whale Trail, each site has a page on our website, and many of our sites have signs. Our first signs and markers went up on the Strait of Juan the Fuca in 2010, and you can see there that's a sample of our interpretive panel and then our marker on the right. In 2011, we had a partnership with Washington State Ferries. We had 2 signs put on every Washington State ferry, 1 at every terminal, and our reach went to over 22 million people each year. The exciting thing about this is that we're reaching a diverse and broad general public. Environmental organizations always have the issue of not just preaching to the choir. How do you reach people who won't seek out this information? With the Whale Trail, we've hit a really sweet spot. These are people just out for a stroll, or they're riding a ferry, and they read this, and it's incredible to watch them to read a sign, walk over to the edge of the ferry railing and look over, and you know something has happened. You know, a light has been turned on about the water that they're passing over. In 2013 was the next big milestone. We had our first sign on the Pacific Coast at Kalaloch Lodge. That again was a collaboration with Olympic National Park and the Olympic Coast National Marine Sanctuaries. And then in 2014, we achieved one of our major goals of extending the whale trail throughout the southern resident Orca Range with our first sites and signs in California, Point Reyes, Point Lobos, and the Santa Cruz Lighthouse. That was part of a tour we did with Eric Hoyt called Orca Tour 2001-4. We also went beyond the southern resident Orca Range. We have a sign in Cambria and San Simeon, California, so we officially extended outside of southern resident territory. This is an example of the sign at the Santa Cruz Lighthouse, and this is what the panel looks like. So you'll see there's a theme. We actually work really closely with our local site partners to customize this according to the messages they want and the look and feel of the signs. We're actually more concerned about the overall message than the specific graphics of it. And in 2015, as Bailey mentioned, we are so excited to start working with the planning team in BC and also at the same time one in Oregon. So this is the Pender team showing you where that Thieves Bay sign is going to go. And then this is an example of the whale trail marker in place. It's a little too small to see, but you can see it encourages people to, lets them know they're at a whale trail location and encourage them to contact the BC Cetacean Sighting Network if they have a sighting. So our goals this year are to fully extend the whale trail from BC to California, following the seeds that have been planted. And now we're getting really ambitious. Since we're going from California to British Columbia, why not go from Baja to Alaska and connect the entire West Coast in this deeply local but widely shared experience of marine mammal appreciation from shore? And we've gone from the southern resident territory into the gray whale migration route. So I encourage you to join us. It's an emerging project. We're evolving as we go along, and it's tremendously exciting to be part, and I couldn't be prouder than to be part of the Whale Trail BC. Thank you. Oh. Questions? No questions? Any questions?
So Lauren, come down. There she is. And she's going to talk about one of the darlings, the latest darlings of the Vancouver Aquarium.
Hello, my name is Lauren Hartling. I'm one of the interpreters at the Vancouver Aquarium, giving you a wave from here at the front of this theater. You can't turn it off, can't turn it off. I wanted to start off, first of all, by asking everyone in this room and the room next door, I can't see you next door, but I'm assuming you'll respond, but how many of you have ever heard of a false killer whale before? Yeah, that is the first time I knew what the outcome of that question would actually be. Out of curiosity, who here has heard of the story of Chester at the Vancouver Aquarium? All right, there's some hands that still aren't going up, so I'm going to give you a bit of a brief kind of check into this darling. As we heard, this is the face of Chester. He is a false killer whale, or Pseudorca crassidens. He was found as a 1-month-old baby who had washed ashore on the coast of Vancouver Island, just south of Tofino on Chesterman Beach. Now, he's super lucky. He was found by people who happened just to be walking along the shoreline, enjoying their morning, and they saw this thing that shouldn't be there. And luckily, they did the right thing. They called the Department of Fisheries and Oceans Canada, who in turn contacted the Vancouver Aquarium Marine Mammal Rescue Center. Now, he was in very serious condition when he was found. He was very weak, emaciated, would not have survived had he been left out, as he was only 1 month old, and as you can imagine, typically animals when they are 1 month old, quite helpless. So in Chester's case, he was taken to our rescue center. We have some rescue staff even in here who worked with him, He has gotten incredibly strong and healthy. Now, Chester is an interesting story because he's what we call a non-releasable animal, meaning he cannot go back out to the ocean. He would not survive. And that's a determination made by the government. The Vancouver Aquarium does not make that decision. That's the government looking at all sorts of data saying, you know what, this animal is not a good candidate for release. So he was deemed non-releasable in May. And then in June of this year, he was moved into one of our main habitats as the Vancouver Aquarium was deemed a place that would be able to house Chester. Now, with all that being said, basically myself as an interpreter, my coworkers, we get paid to talk. We love it. It's kind of an awesome job. In preparation of talking about Chester, we essentially had a year to do all sorts of research on false killer whales. Has anyone here ever done research on false killer whales in particular? A couple of hands are going up. All right, I'm not going to make myself look silly then. I won't go into too many details, but essentially, we looked at every single paper possible. We read everything we could, lifespan, diet, where they live, vocalizations, everything. And we got super excited, and we were so prepared to talk about this incredible animal with our visitors once they got to actually see him. Now, maybe I had overplanned, or maybe I hadn't necessarily prepared myself, but in fact, a lot of the common questions we got from people were not what types of range of frequency of sound or the false killer whales produce vocalizations, or how deep can they dive, and how many different fish are they getting while they're down there. The most common question we got was, why do you call him a fake killer whale? Legit, people actually thought that we, the Vancouver Aquarium, had named him a false killer whale. And so a lot of our conversations ended up being, what is a false killer whale? Hence the title of this talk. It was quite interesting for us to be able to go through and try to have these conversations with visitors who had never heard of a false killer whale before. And back to the question I asked you at the beginning, how many of you had heard of a false killer whale? Well, when we do a talk to about 200 people, maybe 2 hands go up. So you actually have a whole audience of people who have zero knowledge that even something beyond a bottlenose dolphin exists in the ocean. So it's really interesting for us as interpreters, taking researched information that you yourselves have spent your lives collecting and trying to find a way of engaging our audience with that information. Now, most of the common questions we get are, what are a false killer whale? Why do you call it a false killer whale? Which actually led to some interesting research on my own part about finding out that it was in 1846 was a subfossil of a skull that was found that looked kind of like a killer whale's skull, but not quite. So I guess the researchers were like, hey, let's call it a false killer whale. But it wasn't until 20 years later that an actual live specimen was found. So having that information actually helps to answer that question for visitors that, yeah, sometimes names that animals have don't necessarily make sense. And luckily there is a hashtag that came out. Keely showed it to me, hashtag stupid common names. Some of you as researchers might be able to appreciate that, trying to explain why a fur seal is not actually a seal, but a sea lion. So essentially, what it comes down to on our side of things at the aquarium is absolutely, we want to be able to connect people to the animals that are at the aquarium so that they themselves will feel inspired to protect these rare animals, a creature that is rarely seen on the coast of British Columbia. And in fact, in the case of Chester, the last time false killer whales were seen around BC was about 10 years ago. And even when he was found, as far as I'm aware, there were no sightings of false killer whales in the area. Now, we still don't know too much about his origins, but it seems that he does not hail from the Hawaiian, false killer whales. There's a possibility maybe he's from California, but we actually don't know. But this is an incredible opportunity for more research to happen with the help of an animal that, if it wasn't for people walking along the shoreline, we wouldn't actually have. So Chester, living at the Vancouver Aquarium, really helping to open up conversations and hopefully opening up more opportunities for research. But for ourselves as interpreters, really mastering how to connect people with something so rare was a bit of a fun opportunity this summer. And now we are getting folks coming in saying, yes, I've heard of Chester, because he's kind of a YouTube sensation as well, playing with the rain and just being all over cute. Especially now he's only 1.5. He's got a lot more growing to do and a lot more experiences for us to get to share with all sorts of visitors. Have you seen Chester yet? Only a couple of hands are going up, folks. You got to come to the Vancouver Aquarium. There we go. Come to the Vancouver Aquarium, check them out, and help us share the story of this incredible whale, and we'll further some more information on this species as well. Thank you so much. He's cute. That's kind of where you can leave it at.
Any questions for Lauren before we break?
Current weight and length. Oh, I tried to get that information before I got here. He's roughly 9 feet long. When he was moved on June 25th, he weighed about 450 pounds. He's been in this habitat now for about 4 months, 4.5 months. I believe he's around 500, but we really don't have a way to measure him right now weight-wise. We're working up towards that of a trained behavior for him to get to weigh himself. But we do take regular length and girth measurements, and he seems to be upwards of about 9 feet long. Yeah, conversion.
So, as usual, we are exactly on time. So we're going to take a 30-minute break. I only have one announcement, which is this is your last chance, if you're interested in the pizza evening, to pay Aaron, who will be, I think, at the reception table, willing to take your money. And then there is a pay-as-you-go bar. So that's Lusha. Any other announcements? Nope. Please be back at 3:30. Thank you very much.
By the way, we stole this picture from Joan, so thank you, Joan.